Now let's talk about MHC II, which we've used to refer to the antigen presentation on the surface of professional antigen presenting cells. MHC II molecule presents to CD4 positive helper T cells. In reality, MHC II actually refers to the region on chromosome 6 that encodes for the proteins, the alpha and the beta chain protein of this antigen presentation molecule, which is sometimes also referred to as the HLA class II. So in the book, you'll see it referred to both MHC class II or HLA class II. Either way, we're talking about these two proteins, the alpha and the beta chain, that join together and load peptide in endocytic vesicles and present those peptides to CD4 positive helper T cells. So let's look at the genes that encode for these proteins, right? And so it turns out there's, again, gene families, a number of different genes that encode for more or less the same function of protein, antigen presentation to CD4 cells. So we're going to see the concept about isoforms, different versions of the HLA class II complex. So the first version we're going to talk about is HLA-DP. So the HLA-DP molecule, right, the alpha and the beta chain, it's got to be encoded by a protein, a gene that makes the alpha protein and a gene that makes the beta protein. So if you look in the genome, you'll find HLA-DPA1 and HLA-DPB1, right? And these genes encode for the alpha protein and the beta protein that's found in the MHC II molecule. Right? So you again have different alleles or allotypes of these, and some of these are again polymorphic, like HLA-DPB1. There are 114 different allotypes of that, so it's pretty highly polymorphic. Right? For the HLA-DPA1 gene, actually there aren't that many versions of this gene or alleles. There are actually only 15, so maybe you refer to this as polymorphic. There are a number of different ones. Your book uses another term, oligomorphic, which are a handful of versions. And you could refer to the 15 versions as either oligomorphic or polymorphic. But we'll refer to it, I know your book refers to it as polymorphic, so we'll just talk about that. So let's say, for example, let's number these alleles. Let's say you inherit on one of your chromosomes 6, DPA1 version 1 and DPB1 version 20, and then the other chromosome 6, DPA1 version 14 and DPB1 version 77. Right? And so those would encode for proteins. And so typically the proteins on one chromosome, the A and the B gene, make proteins that join together in a complex. So allotype 1 and allotype 20, when those proteins would be encoded for, transcribed, translated, they would join together in a complex. And the allotypes on the other chromosome, when they're transcribed and translated, they would also join into a complex, the class 1 and the class 2. You typically don't see, let's say, for example, here, version 1 joining with version 77 or version 14 joining with version 20. They usually are inherited together and they interact with each other specifically. So both of these can present peptides, right? Which peptides they can present? Well, it depends on the peptide binding motif of each of these. Depending on the amino acid sequences of these proteins, they can interact with different peptides, and they can present different peptides. And again, this brings us back to that concept we saw with MHC I, where the more peptides we can present, the more likely we are to be able to defend against this infection. Remember, these peptides are probably coming from things that we are endocytosing, like viruses, like bacterium. We break down these pathogens, we take peptides extracted from them in the endocytic vesicles, we present these peptides to CD4 positive helper T cells. The more peptides we can present, the more different ones we can present, the higher our chances that we will find CD4 positive helper T cells with a very specific T cell receptor that recognizes this peptide. So the more different peptides we can present, the better. So that's why we have lots of different isoforms, as you will see, of the MHC class II. So both of these are HLA DP molecules, but they're going to probably present different peptides because they have different peptide binding motifs, which we'll talk about in the next slide. There's another set of genes that encode for HLA class II molecules, and we call these the HLA-DQ-A1 and DQ-B1. And so they're going to encode for the DQ isoform, right? And so, again, a number of different versions of these genes that you could possibly inherit, 25 allotypes of DQA1, 66 of DQB1. So if we sort of randomly, assign some numbers to these alleles, let's say you've inherited, allele 10 and 20 of A1 and 40 and 60 from B1, right? And so you will make these proteins, right? And again, we say usually the ones that are inherited together join together and form this HLA2 complex. So DQA1 will form the alpha protein, DQB1, number 40, will form the beta protein. Right? And same thing with the allele 20, that will form the alpha protein. Allele 60 from the B1 forms the beta protein. Right? So you end up making two different isoforms of the HLA-DQ. Each of them can present different peptides. If their anchor residues are different, then you're going to be able to present different peptides. The more peptides, the better. So that's the DP and the DQ, and the third major HLA isotype is called DR. But there's something a little strange about the DR gene family, and so there's an HLA-DRA, and there's a DRB1, and there's another DRB, either numbered 3, 4, or 5. So people actually inherit typically two versions of the DRB gene, BR1, and then another version, either 3, 4, or 5. So that means when you make your HLA-DR isotypes, or isoforms, I should say, so you're going to have an alpha protein made from the DRA gene, and actually there are only two allotypes, so this is not polymorphic, it's oligomorphic, or you could say it's dimorphic. So there are only two versions of this gene, two possible alleles. Let's say you inherit 1 and 2. So you're going to make the alpha protein, right, from that allele, and then what are you going to make which one of the beta proteins are you going to make from the B allele, right? It turns out there are 466 DRB allele, 1 alleles, and then anywhere from 7 to 40 DRB3, 4, or 5 alleles. Again, those are separate genes, and people typically inherit either DRB3 or DRB4 or DRB5. They're located in different parts of the genome. So people usually inherit one functional one of those, and the other two are non-functional. So that really means that you can form different HLA-DRs, a number of different ones, because let's say you form this isoform at the bottom here, this alpha and beta chain, and that beta protein comes from that DRB1 gene, right? Or you can form another complex using the alpha chain and the DRB3 or 4 or 5, depending on which one you've inherited. So you actually can form two different isoforms just from that one chromosome. So those are going to present most likely different peptides. Same thing goes for the other chromosome. So on the bottom chromosome, you can form isoforms using the DRA, so I've numbered it allele number 2, and then the DRB1 and the DRB, I've run out of room there, 3, 4, or 5. So in fact, for HLA-DR, you can make 4 possible different isoforms of the DR MHC class II, which means those are hopefully 4 different peptides, peptide binding motifs that you are presenting. And so if you look, all of the HLA class II isoforms that you can possibly make and present on the surface of your cells, and remember we're talking about professional antigen presenting cells, these are the only cells that make all of these proteins. So these are dendritic cells, B cells, and macrophages, right? And so those 3 cell types take things in, viruses, bacterium, using their phagocytosis or receptor-mediated endocytosis, process those peptides that we've talked about before, load those peptides onto their HLA class II or MHC class II, and now we could see that there are in fact 8 different versions or isoforms that each cell can make. So cells are taking all those peptides that they're breaking down in those endocytic vesicles, they're loading them onto whichever one of these will load that peptide and sending it to the cell surface and showing it the CD4 cells. So again, the more peptides you can present, the more likely you are to find a T cell that matches. And so here, this is MHC II. So those are the genes that are involved, and those are the proteins that are made, and now you can see there are in fact 8 different versions or isoforms of MHC II found on your presenting cells, professional antigen presenting cells.